Mic test. Mic test. Mahina.
Yes, sir, we can hear you.
I'll just include it in my list for. Rerum naturally. So it's uh, doctor for natural science, chemist, it's a German system.
Hello, good afternoon, everyone. Welcome to this talk. I don't know what's the, I lost count, but this is one of the MSI at 50 lecture series talks. And it's my pleasure to introduce the speaker for today's lecture, Dr. Irene Rodriguez. Um, so I think Irene, just like most of the faculty members of MSI, spent part of her career as an RA of MSI, I think with Dr. Koch, Dr. Edna Ganson Fortes. But she's a BS and MS chemistry graduate of Institute of Chemistry here in UP Diliman, and she was also an instructor there. And she got her PhD from Austria with this German title, um, Dr. Rerum Natalareum. I'm not sure if I pronounced that correctly, in chemistry. And then she also had a stint, postdoctoral stint at in Taipei in Academia Sinica. And that's where you develop your expertise and interest in metallomics. Is that correct? Yeah. So this is a brand new field, and it's very exciting to hear, the developments in this new science that is new to me. So, Dr. Rodriguez.
Thank you, Roy, for the kind introduction. Good afternoon to all. Chill lang kayo where you are. And those joining us via Zoom, good afternoon to you all as well. So for this talk, I'm going to give you a walkthrough of why I do what I do and why we pursue the research interests that we are pursuing in the lab. So that's aptly titled The Black Box Pertaining to the Journey if you're checking out the black box of a plane, but hopefully not after a crash. Okay, so this will be quite a journey, starting off with my undergrad to my postdoc stint and giving you an idea again, the platform for the research that I am pursuing in the lab. One moment. Okay, so let's start off with, so I did my bachelor's degree in chemistry from UPLB. So my interest in research was actually seeded aptly in the International Rice Research Institute, where I did my bachelor's thesis research. Okay. And if you are familiar with me and how obsessive compulsive I am, you'd probably think that it's all planned, but no, it wasn't planned at all. Okay, so I was just living my life as an undergrad chem student at UPLB, and then it came to the point that I had to decide to do what kind of research topic I should pursue for my bachelor's thesis. And I happened to come across my professor in organic chemistry, and I enjoyed my organic chemistry, Dr. Heidi Hernandez, surrounded by about 5 or 6 of my classmates that time, and they were asking her questions about what topics are being done in your lab and so on and so forth. And I was passing through, so I stopped and I said, "Ma'am Heidi, reserve a slot for me." And then I left. And then a week after, I got an email from her saying, "Irene, when will you start working?" So I thought I'm going to work, being an organic chemistry professor, I thought that I will be working in organic synthesis and have a run with this named reactions in organic chemistry, having the green yard or whatever else. Yeah. But then when I started working with her, that's when I realized that, okay, I had to work on profiling volatile aromatic compounds in rice. And then I learned after that I had to cook 3 grams of rice in a 3-neck round flask. Can you imagine 3 grams? That's not even enough for, 1 bite. Okay, so 3 grams of rice, and I had to use an oil bath to cook the rice because water bath will not control the temperature well. An oil bath I'll be able to control the temperature increases well. So within 1 to 2 degrees change, I can control that well. And then while cooking the 3 grams of rice, I had to flush high purity nitrogen in the headspace of the rice being cooked, collect whatever volatile compounds are released by the aromatic rice, and then this Tinax trap is a very, I would say, specialized resin for trapping volatile compounds. I had to bring the Tinax trap to the International Rice Research Institute and then process it using their gas chromatography mass spec. During that time, I haven't done my mass spec class, and I was, I think, 19 years old during that time, and I am expected to handle an instrument that is ×2 in millions of my, and then in millions of my age. So the GCMS I was handling was about 40 million. And to me, that was difficult to fathom, 40 million in pesos during that time. Okay, so in front of this, handling this equipment, I encountered several problems with it. So there was a point in the research that I had to participate in opening the equipment and then troubleshooting the GCMS. And for me, that was fun. And I guess that's where I decided I wanted to do research with and work with sophisticated equipment. So lo and behold, I was extended for a semester. And then I was able to identify 2-acetyl-1-pyrroline. That's the compound on the upper left. And this is present in very minute amounts in rice. But even at very minute concentrations, it was very potent. So the rice that you are smelling, jasmine rice from, the Thai rice, the ones that are smelling really nice, definitely they will have minute amounts of acetyl-1-pyrroline. Yeah. So I did this and then finished my bachelor's degree, and I was thinking, okay, what to do next? So Mom Heidi was gracious enough to offer me to continue working for her. And I thought I was going to continue working on rice, but no, I started working with ilang-ilang, doing the extraction of ilang-ilang, working again using a different equipment. So I was introduced to a supercritical fluid extractor, and during that time, the extraction using supercritical fluid extractors using carbon dioxide from the air was the in thing. So it was environment friendly and so on and so forth. We're not using hexane or carbon tetrachloride and all those organic compounds that we would like to avoid. Okay, but after a while, my mother asked me to rest. So I went home, and then after about 3 weeks of not doing anything, I was bored. And then I decided with some friends, okay, let's find work. And we prepared CVs and explored UP Diliman. And that's where I was introduced to the UPMSI. So I worked as an RA under Dr. Edna Ganson Fortes. And as a chemist, I asked Nanay Ed, why did you hire me? Because my work on seaweeds was on the reproductive biology and on the ecology, the biology of Gilligella acerosa, a very good source of agar. And during that time, her major work was on collecting pregnant Gilligella acerosa from the field, and then having this brought to the lab. And in the lab, the spores are released, and I think Helen will know more. Helen Dio just arrived. She was my teammate in all the field works that I did in Corimao, Ilocos Norte. So we were collecting pregnant Gilligella acerosa and then monitoring the growth from the spores under the microscope here in the lab, where the seaweed people are still working in. Okay, so this was all under the microscope, and I was able to grow them to, I think, about a centimeter long, and cleaning them under the microscope every day of my life. And after that, I thought, okay, microscopy work is not for me. And I decided, okay, I'd like to pursue my master's. So I enrolled in the Institute of Chemistry in Diliman, still working on Gilligella acerosa. The good thing about this is I was introduced to cell cultures. And that's going to play very critical aspect in the work that I will be doing in the succeeding years. So being at the Institute of Chem now, so I transferred to the Institute of Chem, but the influence of MSI was already deep, because when I decided to pursue my thesis topic, I knew that I wanted to do work in water, related to water. So when I decided for a thesis topic, the one that was available for to me that time was looking at the chlorination disinfection byproducts of drinking water, working with Dr. Maria Pythias Espino. And doing this work, I was introduced to the entire process needed to have our drinking water available to us in the tap. and the health risk associated with it, and all the other problems, problems in regulation, in implementation, monitoring, and all of those things. So it had the social aspect, it had the chemistry in it. I was once in a conference presenting my results here, and I was asked point blank, "You're saying that that chlorination is causing a lot of these disinfection byproducts, and the disinfection byproducts are cancer causing, but how are you going to balance it off with problems in, for example, waterborne diseases?" Because frankly, until this time, the best disinfection disinfection process for our drinking water is still chlorination. So we just have to balance it out. Not the highly technical ones and the sophisticated ones of treating our water is not always available. Okay, so in highly industrialized countries, then they can opt to have reverse osmosis. But for countries like ours, especially in the rural areas, it's still chlorination that will offer the best protection against waterborne diseases. So here I was working with on these compounds, looking at production of this chloroform, bromoform, the chloroform in drinking water as a result of the disinfection process. So if you look closely, all of these are emanating from my undergrad years working on organic compounds. So when I finally decided that I'd like to pursue a PhD or doctoral studies, I was adamant to move away from organic analysis. So for my doctoral, it was a conscious decision that I'd like to work on trace metals, and that's where when I was offered an opportunity to pursue it with to do doctoral studies at the Karl Franz University of Graz in Austria with Dr. Walter Gessler, I did this. So arsenic analysis in various biological and environmental samples. And working with Walter, his lab is, if not the best arsenic lab in the world, one of the best. So they are known the lab is known for arsenic research. Okay, and working with him, I was introduced to the concept of speciation, that the toxicity is defined not by total concentration of metals, but by the species of the metals at which they are present. Okay, so inorganic arsenic is the one that is toxic. The organoarsenicals, such as arsenobutane, arsenocholine, which are present in very high amounts in seafoods, the organoarsenicals are non-toxic, not toxic at all. So the non-toxic forms are the ones that are present in seafood, so fish and other shellfish. Okay, that is to say that knowing the species concentration in terms of monitoring metals for health reasons or for safety, we have to know species. It's not enough that you know total arsenic and then you just ban it. Okay, but that is not true. For example, if you're looking at mercury or if you're looking at cadmium or lead, because cadmium and lead, regardless of whatever form they are present in, they will always be toxic. Okay. So I'd like to share this with you because this is also one of the things that was very important to me. So the Paracelsus maxim, again, the dose makes the poison. So I'm presenting it here in the German language because it's quite cute. If you check it out. So in German, alles Ding sind Gift und nichts und Gift. Allein die Dosis macht, dass ein Ding kein Gift ist. Gift being, in English, poison. So please be careful when you're giving your gifts. Okay, especially this time around. It's almost holiday season. And Yeah, but we choose our poison, right? Yeah. So having that kind of mindset and being deep into the metals. So that's when I started working in the field of metallomics. So metallomics being looking at studying the total concentrations, the speciation, and the cycling of trace metals in the environment, and also in biological systems. So when I returned to the Institute of Chemistry, I was working on trace metals. We had projects on trace metals in drinking water, trace metals in Laguna Lake. So we were collecting fish, we were collecting sediments and looking at, and also the water, and we were tracing if cadmium, lead, arsenic is correlated in these different matrices. And I had a student who invited me to go to Benguet, and we requested mothers to give us breast milk samples, and we were also monitoring different metals in their breast milk. Okay, the the village that we went to in Benguet was very close to the mining sites, the ones that are not properly managed. So you can imagine that it was very difficult for us to approach the community. And we specially selected participants who have not gone out of the village. So they're staying there. But that was a bit heartbreaking, collecting or asking breast milk samples from them, because they were carrying their babies, and they were pumping on the left or on the right, and the babies sucking from the other nipple. So it was during that time, we're not yet required to submit this ethics review and such, but personally, it was a bit. I was disturbed at I would say, at the end of the sampling. But then again, as scientists, we know that these things are important, especially if we want to look at to work on epidemiological studies, looking at long-term effects of trace metals on the population. Okay, so working on different aspects of trace metal cycling. And then I was at that point that I wanted to do epidemiological studies in the Philippines, but it was difficult to get funding. So I was thinking, okay, probably I need more expertise. So that's when I decided I want to pursue a postdoc. And I met my postdoc supervisor here in the country. Academia Sinica sent a delegation to UP Diliman that time, and then I met Tung Yuan, and then we discussed. She said, "Come to Academia Sinica, do a postdoc with me." So I came to Academia Sinica, really thinking that I will continue on working with the metallomics aspect that I'm familiar with, looking at trace metals as pollutants. But then when I joined this lab, he was doing all of these crazy algal cell cultures with cyanobacteria and a lot of other organisms, phytoplankton. And they were going out in the ocean collecting water samples and doing in situ enrichment studies of metals in trichodesmium. So one cyanobacteria species that's able to fix nitrogen. So it's a diazotroph. Okay. And lo and behold, I had to go back to my time in MSI, where I had to do algal cell cultures, macroalgal, but this time around I had to do microalgal cell cultures, totally different way of doing things. I had to do batch cultures, and from there I was trying to recall, do I still remember my photosynthesis and all the electron transport cycles and all of those things? So yes, coming from a very almost rigid analytical chemistry background, with all of this, we're thinking about epidemiological studies, and there I was being thrown in doing algal cell cultures, and voila, microscopy work. So for 3 months, I was just in front of a microscope doing comparative studies of using a Coulter counter against a microscope in counting cells. And if you check it out, there are filamentous. So counting the number of cells and against the cell volume and all of those things require a lot of work. So for those doing microscopy work, I really salute you in your patience to do this. Yeah. So I got very interested in trichodesmium because for cyanobacteria, they're quite cute. Okay, so photosynthetic organisms that are able to do nitrogen fixation, they have ways to manage those two processes at the same time. Okay, so photosynthesis, when organisms carry this out, they are releasing oxygen in the environment. The oxygen, of course, when it gets across one lone electron, then it will just form a superoxide. The superoxide, even the oxygen, will deactivate the nitrogenase enzyme that's necessary for nitrogen fixation. So for majority of photosynthetic organisms that are also diazotrophic, what they do is the spatial separation of the two processes. For example, you have Anabina here. So you have the heterocyst for nitrogen fixing activities, and the vegetative cells are the ones that are used for photosynthesis. Okay, so spatial separation of the two processes. They are connected, of course, because these are mostly filamentous. And other organisms do it in a temporal separation of the two processes. So during the day, photosynthesis, and then they store whatever carbon they can fix, preferably glycogen. And then the carbon reserves are utilized at night for nitrogen fixation. Okay, so bear in mind that nitrogen fixation is an energy-intensive process, so it will require a lot of energy, whatever energy the organism can spare. The thing with trichodesmium, and that's why I am so enamored with it, yeah, is it can do both processes at the same time in the same cells. Okay, so the proposition now is it is able to separate the two processes in a spatiotemporal way. So it's kicking in photosynthesis and then switching that off, doing nitrogen fixation, and then photosynthesis again, just to ensure that it has the energy required for the nitrogen fixation. Then what my supervisor is proposing is that the organism having the machinery to produce a nickel containing superoxide dismutase, that the nickel SOD is protecting the nitrogenase enzyme from the activation, even with the production of oxygen during the photosynthetic activity. So essentially that is laid out here. So photosynthesis releases oxygen and other reactive oxygen species. The nickel SOD that is present in the organism is now converted to hydrogen peroxide, and that is converted by an iron-containing catalase into water and oxygen. Okay, so with this, what's critical will be the nickel availability. So to prove that point, they carried out, of course, this is how we do algal cell cultures under trace metal defined conditions. So all of your preparations has to be done inside the clean room. So we will have a clean room, something similar to this. It's actually here already. So this is Hannah Reich, one PhD student that I supervised while I was in Taiwan. So she's preparing her batch cultures. So that's how we do batch cultures under temperature controlled, light intensity controlled, and humidity controlled growth chambers. And all the manipulations has to be done in a class 100 trace metal clean room. Okay, so we carried out these experiments before I arrived in Taiwan. So on your y-axis will be biomass curve. So this is cellular volume in cubic micrometer per ml of the cell culture. And on the x-axis will be the growth time in days. And the blue ones represent high nickel treatment. The pink ones represent low nickel treatment. The one in the left is your low light treatment. The one on the right is your high light treatment. Okay, so from here, from the growth profiles itself, there's proof that indeed with high nickel availability and high light, trichodesmium erythrium is able to perform well, to grow well in terms of cell density. And when I joined the lab, I repeated this, but in addition to that, to the growth profiles, I looked at nitrogen fixation activities using the acetylene reduction assay. And I was able to show that in highlight treatments with high nickel availability, the fixation of nitrogen is higher. So higher nitrogen fixation rates are observed in those conditions. Under highlight, high nickel availability, as opposed to low light and low nickel availability. Okay, so with that, I was kind of curious. What else? So sorry about this. I cannot find this figure in color. So this is from Berman Frank in published in 2001. So I will sort of we proved that the SOD can be important, but only based on availability and intracellular intracellular nickel content. But what I'd like to know is how the iron nickel uptake hydrogenases come into play. Okay, so nitrogen fixation being a energy intensive process, theoretically, if an organism is fixing 1 mole of nitrogen, it's also releasing 1 mole of hydrogen in the process. Okay, but in reality, that doesn't happen. So it's 3:1 or 4:1. So 4 moles of fixed only releases 4 or 3 moles of nitrogen only releases 1 mole of hydrogen in the process. So we are thinking there's another nickel containing enzyme that is perhaps affecting the process there. And we're looking now at a nickel iron uptake hydrogenase. So the function is, of course, once your hydrogen is available in the surrounding environment, then it's just taking up that H2 and converting that to H+. There's another type of hydrogenase that's present in most organisms, and that is a bidirectional hydrogenase controlling the proton, and so H+ or H2 in the system. Okay, so if one is higher, then the bidirectional hydrogenase will kick in to keep the equilibrium between H2 and H+. Okay, so now going back to this, we then carried out several treatments. So again, that's your cell volume on the y-axis and duration of the experiment or incubation time in the x. Okay, so on the left will be your growth profiles under various iron and nickel conditions. Okay, and in the middle one, that's your nitrogen amount of nitrogen fixed. And then we relate that now with the hydrogen produced or liberated by the different treatments. Okay, so very important to note here, the highest nitrogen fixed are from low, okay, so I'm just saying low iron, high nickel treatment. Okay, so essentially the high nickel treatments gave us the highest nitrogen fixation rates, which goes back to our hypothesis that nickel SOD is important. But the highest hydrogen produced will be from treatments with high iron and high nickel. Okay, so we're wondering, well, what is this? And if it's not yet obvious why I am interested in the hydrogen, it's because of the potential biotechnological applications. Because you have a marine organism, it's photosynthetic, and you can just imagine the possibilities for this. If you can grow this in larger quantities, that can be a source of clean energy. Okay. So to prove that point, we did immunocytochemistry and to check out if indeed the nitrogenase enzymes are expressed in the same locations as the photosynthetic enzymes. Okay, so for photosynthesis, we're just looking at D1, and from initial results, we are observing that they are co-located. So indeed, you have nitrogenase, you have photosynthetic enzymes being expressed at the same time as nitrogenase enzymes. So co-occurring two processes in the same cells, even if they are not compatible processes in the first place. And I was adamant, there's no existing antibody for nickel SOD when I started this work. So I contacted one company, gave them information, and then I asked them, please prepare me the antibody for the nickel SOD. And you're actually looking at the first results proving that indeed Trichodesmium erythraeum has nickel SOD. Okay, and I was so happy with this work. But if you note there, that's manuscript in preparation. That's a task I haven't ticked off in a long time. Okay, so to prove a point, nickel SOD is indeed expressed in Trichodesmium erythraeum, and what we're showing here is indeed it's important in both the nitrogen fixation of Trichodesmium as well as photosynthesis. Okay, and then we checked out this other cyanobacteria species. This is Cyanotisi now, unicellular. It does photosynthesis during the day and nitrogen fixation at night. And we also looked at the influence of iron and nickel availability. And as you can see here, this is now low iron and high iron treatment on your x-axis, and on your y-axis, the one on the lower panel will be your nitrogen fixation rate. The one on top is hydrogen accumulation rate. And you can see here that there's no correlation between the nickel treatment with in terms of nitrogen fixation and hydrogen production. So what we think happened here is the role of bidirectional hydrogenases. So for this particular work, since cyanotisi is quite abundant especially in coastal environments, it has a lot of biogeochemical implications, because you may use the hydrogen concentrations present in the water to also denote nitrogen fixation activities in the area, because it's so much easier to monitor hydrogen concentrations rather than nitrogen production using the acetylene reduction assay or N15 enrichment studies. So that's a major application of this work. So this is now about the work in cyanobacteria, and then I have 30 minutes. Yeah, I'm just going to go through this quickly. I also did work on Zooxanthellae because there was one afternoon that Tung Yuan came from Xiamen University, and he brought Fugacium kawagutii to us in the lab. And I was crazy about Fugacium kawagutii because it has all 4 types of SODs. So iron, manganese, nickel, copper, zinc, SOD. So it has all 4 types. And I did some work on the trace metal requirements of Fugacium kawagutii. And from here, this was the first work ever on trace metal requirements of Zooxanthellae, the ones that are symbiotic with different types of cnidarians. And what this paper did was set up the foundation for a lot of other works on metal-containing enzymes in Symbiodinium kawagutii. I just like to point out that when I did this work, if you check out the panel here, again, this is cell density and this is duration in days. The treatments that I did, I had a control with all trace metals, and I did treatments without nickel, without manganese, and without copper zinc. I did not do without iron. I did that in a different experiment. Okay, so if you check out the bottom panel, I did combinations of the metals. So I had treatments here without nickel manganese, without nickel copper zinc, without manganese copper zinc. And this was, on hindsight, a very poorly designed experiment because I was observing differences in growth profiles. I was observing differences in their intracellular metal content, but I'm not able to ascribe that to a very specific growth factor because I did combinations. Okay, so I repeated this experiment, and when I repeated this experiment, I did it in such a way that I did treatments without copper, without manganese, without nickel, and then from previous experiment, I was observing that if I don't supply it with zinc, it's not going to survive. So I just had 1/5 zinc concentration, and I also had a treatment with just 1/5 of the iron concentration. I learned from my from the limitations of my previous experiment. And then in addition to monitoring growth profiles and all of those things, I also asked collaborators to do differential gene expression. And what I'm showing here now are the identified, they were able to, so check out the paper here from Xiamen University with Singeline. So they were able to identify 372 genes that are affected by all four the limitation of all the different in all different treatments, and most of which pertain to, now this is the size of the circles will be corresponding to the gene number, and the color, the warm colors represent the Q value. And majority of the 372 gene number that they were able to identify were related to all these processes. So the metabolism of pyruvate, propanoate, nitrogen, and all of those things. And moving forward, these are now the differential gene expression in terms of the changes in fold changes, as well as identified the specific processes that were affected. So we're very happy with these results because indeed, okay, we're able to find out that if you supply this in limiting concentrations, those are the processes that will be affected. Disclaimer, again, these were in batch cultures. So the kind of conditions that the organism is subjected to is under very, so if it's under batch culture, so it's only pertaining to that. It would have been better if I'm able to carry this out under continuous culture conditions. But from here, a lot of information can be derived, and you can think of the possibilities of follow-up experiments that can be done. Okay, so with that, I had this thought. So this brilliantly summarizes the role of trace metals in photosynthetic systems. And this is now the thing that I would like to pursue. So when my postdoc stint was coming to an end, and I was deciding to return to MSI, this was now my blueprint. Okay, so metals in aquatic plants and how it relates into, or how it can build in terms of biogeochemical cycling or biotechnological applications. And then I returned to MSI, and then I found the love of my life and funded the IBR lab. So the Inorganic Biogeochemistry and Biotechnology Research Laboratory, and our logo is this, care of Paulo Calalang. And the logo in itself represents a lot of things. So biogeochemical cycling, that's the interplay with a circular thing that you can see on at the outset. Okay, the green here, that's the the biology aspect of our work, where we consider all photosynthetic organisms, understanding the trace metals. The general part in our work will be something represented here. So you can look at it as sediments falling off, but the way I look at it will be electrons that are ejected from the atomic structure. And the last panel, the last part there will be where the biotech applications come into play. Okay, so in our lab, the first funded project will be from DOST Picard, okay, and also the DOST GIA, focusing on HABs. So why am I working on HABs? Because HABs are dinoflagellates, and I had work on Symbiodiniaceae, which are also dinoflagellates. But this time around, we're looking at the influence of trace metals, both from whatever source, especially aerosol deposition, and looking at how trace metals interact with the major nutrients and control growth, as well as toxin production of HABs. Okay. And because of these funded projects, we're able to build our lab at the BML. Okay, so this is room 205, formerly the lab of Caro at the BML. So thanks so much for that, Caro. We converted the lab into the algal cell culture facility at the Bolinao Marine Lab. Okay, so it's not trace metal clean, but it's semi-trace metal clean lab. So we're able to do a lot of things here, manipulation of trace metal conditions in different organisms. In addition to this, we were able to put in, this is room 203 at the BML. So we're able to put in, we have simple equipment there, but we also have a flow cytometer for cell counting. Because again, you know, if you can avoid microscopy, then do avoid it. And then we have a nutrient analyzer here. It's a Sun++ auto analyzer with modules for nitrate, nitrite, phosphate, and ammonium. And here in Diliman, we built our trace metal clean facility. So this is the former kitchen. It's right in front of this room. It's not opened yet because it's not yet transferred to me. So it's a trace metal clean room. This is the trace metal hood. That will be a class 100 trace metal clean facility. And of course, I continued working on coral, on Symbiodiniaceae, looking at the coral Symbiodiniaceae relationship and the role of the metallome there. So this is a funded project from UP System. And the first experiments that we did will be looking at iron and nitrate interactions. So this is actually the thesis topic of one of my graduate students, JJ Versola. And his results are showing that indeed lower, of course, this is expected, lower iron results in lag of the growth of the organism. So both for Symbiodinium microadriaticum and Cladocopium goroai. Okay. So in addition to this, he's monitoring nitrate changes, ammonium changes, as well as intracellular metal content, the biomolecules produced by the organism, as well as POC, PON content, all of those things. And right now, I'm also implementing this project. So it's the phytochemical characterization of macroalgae for food, and I forgot the entire. It's PhycoPro, co-led with Dr. Roleda. And our component here is on the establishment of the seaweed chemistry laboratory here at UPMSI, and establishing all the protocols for the typical ones that are needed for characterization of seaweeds, the chemical parts. So from here, we are now, this is right at the back of this room. So that's the seaweed chem lab. These are the first few equipment that we have there. So right now we're setting up the CHNSO analyzer. This is a graphite furnace atomic absorption spectrophotometer. Not fully installed yet. This is a high HPLC triple quadruple mass spec. And that one there is a GC with a flame ionization detector, electron capture detector. So it's both a FID and an ECD. Okay. And the rest of the room will be for wet chemistry preparations and one room for dry lab. Okay. So lots of field work. So we're not just lab-based, we're field-based as well. So this is from sampling in Sorsogon Bay for the hubs work. Okay. And this is one experiment that we did at the Gigawanan channel. We were looking at the influence of iron and silicate in the growth of the phytoplankton community structure from samples in the Gigawanan channel. This work was actually inspired by Dr. Fernando Siringen during the cruise last year in the Philippine Rice. We were chatting with each other and said, why not look at the influence of silicate in hubs? So we have yet to process the results from that work. From one field work in Iloilo, and this is my seaweed chemistry and seaweed chemistry, seaweed utilization class. So featuring some people that you are familiar with already. Okay, and during the last PAMs where some team members shared their work. So that's JJ there sharing his work on iron availability and nitrogen utilization by Simbe Dinyasi. This is Dial presenting their work on Tetraselmis tetrahele, and Richard was elected representative to present our work on the hubs. And this is Aubrey for most of you, and Geniel or Jeannie for most of you, presenting our work on preliminary work on the coral diseases, the occurrence of coral diseases in the Philippines. So I also worked with an industry partner, so Magwayan Organics, and we built this facility at the hatchery in BML. So we call it the chemical ecology room, where we can have 48 tanks, where we can control light intensity, we can control the flow, we can control nutrient input in each tank, and the flow rates as well, but not the temperature, because the temperature is just what's provided in the ambient room. Okay, so the work with Magwayan Organics was in collaboration with Van. So this is the Rodriguez's of MSI, with Lon Ligson, Rap Heson, and Jeff. And results from that work were already presented in the recently concluded APCRS in Singapore. Okay, this should translate to a publication very soon. Okay, so aside from this, again, the biotechnology work in the lab, we are looking at microbial cell factories using photosynthetic organisms. The aspect that we are interested in, of course, a lot of work has been done on all other things. We are interested in that tiny thing there, the nutrients and the trace metals, and how trace metals, how we can control that, manipulate that, and change or use it as a platform for production of whatever desirable material. And other than this, we are interested, of course, in the seaweeds and the interrelationship of the seaweeds with corals, because they are always coexisting, and furthering our work on coral diseases. Okay, so now you're wondering, why are they doing a lot of things? Everything is related to the trace metal work that we do in the lab. So we are interested in how trace metals control the growth of these organisms, and how trace metals or the trace metal containing enzymes play a role in the association of corals and seaweeds, and how that leads to diseases. These are photos by Amar Jansi, who was a part of my team last year. And you can see here that the close interaction of some seaweed species with corals. Some of them may not cause any problems, but some of them may cause diseases. So I don't know if it's antagonistic or whatever compounds are being released by the organisms. Those are exactly the questions we would like to address in this particular work. Okay, so further photos. These are photos taken by Bree and Jeannie in KAG when they joined the cruise last year. And this particular part of the work is being pursued in collaboration with, if you can identify them right there, that's Dr. Yu and Dr. Santi Agnes. Okay, so in one trip that we did earlier last month in Caramoan. Okay, so this is a continuing work between the three of us, and if you'd like to be part of the team, we welcome collaborations within the MSI. Now, for international collaborations, I have active collaborations in Austria, of course, Taiwan, because I spent quite a few years there. With the US, UCSD, so FICO-Pro is in collaboration with UCSD. And Hannah Reich, my doctoral student before, is now an assistant professor in SUNY. And other friends from Penn State, essentially this is Tad Lajeunesse. And I have other collaborations that are in the pipeline in Thailand and Malaysia and the UK. So I'd like to acknowledge, of course, the funding agencies, first and foremost, UP System, because of my Balik PhD, and both the Balik PhD support and the support on my research on coral Symbiodiniaceae associations. Of course, MSI for providing all of these, the availability to me. And really the support here at MSI, admin staff and everybody, o, di ba, may drum roll. Really, I am grateful for the support in all aspects. Of course, DOST and DOST Picard and CHED for funding most of my projects. I am thankful to my lab. That's us early this year, January. Meron kaming guest dyan. This is Casey, estudyante ni Rach. Guest namin during the one activity. And I'd like to share this with you, a quote from Plato, that the purpose of education is to give to the body and to the soul all the beauty and all the perfection of which they are capable. I'm telling my lab members always, we don't aim for perfection, we aim for excellence. Because if you always aim for excellence day to day, then you will arrive at perfection. Yeah. And if you're interested, check us out. This is the website of the lab. And if you are interested to work with us, do send me an email and start your own black box. Because, for graduate students there who would like to work with us, would like to work in my team, you may join me as a graphite. You may come out as a diamond. Thank you all.
Well, thank you very much, Dr. Irene Rodriguez. Do we have any questions from the audience, both here on site and Facebook or Zoom? I do have a lot of questions because you bring up a lot of very interesting stuff. So if I may start it, if no one else is raising their hands yet. So it's like we've gone through Irene's life, a very long narrative of her, educational track. It doesn't mean you're that old, but full of experience is what I meant. So, seeing those metallome studies where you have, a lot of metals, iron, nickel, it brings to mind that there are several areas in the Philippines with high metal content. So it seems like you have a really nice playground here. Um, what specific areas in the Philippines do you think you can target with all this metal enrichment and linking it to your laboratory fun fair?
This is rather tricky. I'm certainly interested in areas where there are mine tailings because that's where you get all the interesting photosynthetic organisms. So isolate them from there and then bring them to the lab and then look at the possibilities for them. So how do they utilize it? So down to the cellular level, it's very interesting because that gives you indeed a lot of avenues to utilize the organisms themselves and utilize it eventually for, let's say, bioremediation. So yeah, the nickel mining sites, the copper mining sites. So yeah, wherever you encounter those, that's always interesting.
Questions from the audience? So you mentioned nickel mining sites, and Philippines globally is one of the, we have a lot of nickel ore. In fact, we export a lot of nickel ore. They come back to us as your batteries in your cell phone. Uh, do they, would this like factor in, like in your research, those areas might have a lot of high, that helps the high photosynthetic and the high nitrogen capture? Would that like eventually go in that direction, or I don't know what kind of research we need to do?
Yeah, for not just for biogeochemistry, but also again for bioremediation potential of these organisms. And the thing is, you can actually grow them in ponds, and from there, so it's not just their bioremediation potential that you can harness. You can also make use of the organism to produce specific products. Yeah, so you can tailor, not going into, because we're talking about growing them in ponds. So not modifying them to the cellular or the genomic level, but only modifying the growth conditions with what you expose them to. So that if they are released in the environment, then you don't have a problem because they are from there.
That's
Yeah.
kind of tricky.
Yeah. Well, you're not modifying it in in terms down to that level that it can affect uh, the offspring of the organism, but just modifying the growth conditions. That's how we are approaching this. Because if you are going to use this for bioremediation, again, they are in the environment, and there's a potential of these organisms being released in the environment. So you don't want any changes down to the genome.
Yes, so everybody got that. Also, I think some of the students here and online, what they might have in mind, because you have a lot of cool stuff in your laboratory, a lot of not just the instruments, but also the experimental setup, even in Bolinao. So if they need to use this, how do they, they might not be your students, they might be from another lab, but seeing all those aquarium,
Yeah.
those
Just send me an
instruments.
email, we can discuss.
Okay, you hear that?
I normally answer my email within an hour or two. So
And what opportunities do you have right now? Funded or non-funded scholarship? Or do you need someone in your lab right now?
well,
What topics?
we are welcoming students, we are welcoming collaborators. So if you're interested, shoot me an email and let's discuss.
Anyone want to ask research topics or anything? Yeah.
Hey, Mama. Hello. Hey, Mama Irene. I'm just curious because, for example, there are cyanobacteria that produces natural products that binds with metal. So I'm just wondering how does interferes with your research design? For example, if you're monitoring the trace metals in your for trichodesmium or cyanothiacate.
So the question is whether the organisms themselves or they are producing organic matter that's binding to the trace metals. Correct.
Yes, yes.
Yes. So to address this, we control bioavailability using ethylene diamine tetraacetic acid. So that's ligand exchange. There will be ligand exchange there to ensure that they are bound to EDTA, and then that's how we control the bioavailability. So yeah, so we do this in such a way that okay, you have I did several experiments before where I don't add EDTA. It's just the natural seawater that is available to me, and that's where and then I add my treatments. So increasing concentrations of iron, increasing concentrations of nickel. And you do that, and then you do your treatments with EDTA. And then from there, you can actually see the differences. And the other thing is you have to be very careful when you collect your samples for intracellular metal analysis. You can simply rinse your cells with ultra pure water, but we also have a calcium oxalate rinse so that the adsorbed metals, not the intracellular metals, so only the adsorbed metals will be taken out, filtered, and will not be part of your analyte. So there are checks and balances sort of that you have to pay attention to when you do the trace metal analysis.
Thank you, ma'am.
We have another question if there's no one else raising their hand. So, in Batangas, for example, in Mabini, where we have submarine groundwater discharge, we also found out that there's a lot of metals in it. So, for example, iron is really high, lead is high, arsenic is there. And there's even one paper reporting that there's lead in some of the fish tissue in Mabini. So, your presentation got me thinking again about our study area. So, you mentioned that organic arsenic in seafood is not poisonous. So it's safe enough where we are in Mabini. But then we have the lead, and what can you say about it?
Yeah, once you have lead and it's above the maximum allowable concentration, then avoid eating that food because it is cancer causing and it's deposited in the bones and it has long-term health effects. So I can only say that for arsenic because arsenic being it's actually converted inorganic arsenic in water is converted to methyl containing arsenic by microbes. So thanks to I think it's fucus. So thanks to them, it's detoxified in that particular pathway. But lead, regardless of the form, it's going to be toxic.
Okay, so we need to note that also for those.
The same is true for cadmium.
Okay. Okay, going back to arsenic, because at least that's for the seafood, but then there's still arsenic in groundwater. There's still arsenic that can be accessed not as organics. And how do you propose we look into this?
Okay, so groundwater being taken out, especially for cooking or drinking, we have to pay attention because if present, especially in freshwater systems, arsenic will be present as inorganic arsenic. So either arsenite or arsenate, and it will be problematic because it will act in the same way that we are utilizing phosphate. So that's actually the mechanism of your arsenate. It replaces or it follows the chemical pathway for phosphate utilization, and that's where it causes the problem.
Thank you for that. We have to take account for that. Yes, Dr. Yap.
Helen, just Helen. I am, congratulations and thank you so much, Irene. It is such a great pleasure to just be sitting here and listening to the conversation. I mean, also with Caro here moderating. I hope the students appreciate this great privilege. There are not many institutions here in the country that have this very high level of scientific conversation. Because here at the MSI, and I'm talking, I think I'm not biased, we have some of the best minds in any field of science. I hope you appreciate that. Okay, it's really, I really wish the audience were although I see a couple of guys who I've seen the other day. Now I hope there'll be more who will attend. Okay. Anyway, my question is this: so you want to relate your research to the age-old problem of HABs, no HABs. Now, what I'm thinking is the theater is very dynamic and changing all the time, and in a bad way because we have all these pollutants that are being discharged into the ocean, many of which are hardly characterized because you have all kinds of industries and the chemists are to blame. You are inventing all kinds of new chemicals in the hopes of helping human society. But in food, in medicine, daily life, cosmetics, you're inventing and inventing and inventing compounds. Okay. So the backdrop against which HABs are taking place is becoming, I think, more complicated. But is it already known which is the trigger boom that will cause a bloom? Is it already known? Because, you have these big surprises. Suddenly there's a bloom. And then you have all of this chemical soup, no, in which the dinos are swimming. But then suddenly, boom, there's a bloom. Is it already known which is a chemical trigger?
I cannot comment in terms of macronutrients, but in terms of trace metals, I would say iron and cyanocobalamin, vitamin B12 will be major factors. And with regard to trace metals, we are actually very interested in the influence of selenium because a lot of organisms require selenium for selenomethionine. And in terms of this, that's why we are interested in looking at aerosol deposition, both the dry and the wet, and studying the control of the trace metals on nutrient acquisition, because these are the things that are not yet known for harmful algal bloom species.
I'm just a very simple-minded ecologist, but can an experiment be designed, like even in Bolinao, where you have controlled containers with controlled conditions, and you culture the dinos, and then you add one or more, a combination of these factors you just mentioned as the magical ingredients which will trigger a bloom? Can you test this experimentally?
This is the idea of the Gigiwanan channel experiment that we did. So from the channel itself, we collected around 5 liters of water and then did enrichment experiment. So for that particular experiment, we did iron and silicate. Unfortunately, this particular experiment is not my priority at this time. So I haven't processed the results. I'll get back to you about this, Alan. Uh, uh, No, we did not observe a bloom. So I'm thinking now it might be because of the low volume of water that we used. So I'm actually thinking of using continuous cultures for this. Because the beauty about continuous cell cultures, if you're familiar with it, is you have you have a source, and then you manipulate the growth conditions, the growth parameters in your source. So nutrients and micronutrients, and then have that supplied to a container, and the organism in the growth medium itself will be kept at exponential growth always. And from there, you can, from the outflow, you can monitor whatever parameter. Look at lipid production, look at intracellular content, look at how it's using up the macronutrients you are supplying it with. So there's a lot of potential there. And you can also do a soup of organisms in the growth medium. So you're not just limited to one organism, you can have mixtures of organisms.
So, given exponential growth and all of that, how do you define a bloom quantitatively?
This I cannot answer at this point because I'm not an ecologist. I'll have to discuss this with you.
Thank you. We also have questions online, right? First question comes from Dr. McGlone. She says, iron fertilization became interesting in the past because of its potential to reduce CO2. However, the idea did not take off. Your thoughts on this? Are there other metals that can be tapped successfully for a similar purpose?
Yes, the John Martin hypothesis. That's a very tricky one to do this type of experiments in the field. But actually, I would say this kind of experiments, we are doing it already, not intentional, but rather unintentional with the mine tailings and the metals that we are inadvertently releasing in the environment. And from there, perhaps it's just a matter of collecting samples from these areas and looking at changes in the phytoplankton community structure. So in terms of other metals, we actually were thinking of doing a nickel enrichment experiment, but nowadays it's not allowed to do these kinds of things, especially if you're out in the open ocean. And it's also something that I will not advocate for because there are a lot of other things that we cannot control if you do it outside in the field. So for fertilization studies, releasing it in the environment, we can do this by just monitoring what is already being released in the environment, as I was mentioning earlier, from mine tailings and all. But to do it intentionally, I will not advocate for that.
Thank you po. Our next question po is from an anonymous attendee. Research outside the country tend to have so much focus on carbon sequestration by optimizing the carbon cycle uptake and its downstream utilization pathways. Very little research has been done on optimizing photosynthetic efficiency. If we want to also do research that also aims to do carbon sequestration research, how would we approach this? How many strains of photosynthetic bacteria do you have in the lab? Or would seaweeds be more applicable for such applications?
Yes, I'll answer the last question first. In terms of biomass, indeed, seaweeds or the macroalgae will be more, I would say, it will sound more advantageous. But there are advantages in using unicellular or filamentous photosynthetic organisms. Okay, you have to do a list of the advantages of disadvantages if you will use micro or macroalgae for whatever purpose. Yes, a good point about the us studying the photosynthetic efficiency and that. But for that particular part, I will collaborate with someone else because photobiology in I'm just mentioning photobiology as one aspect of studying photosynthesis is already a very, very complicated thing. Okay, so I also did work on looking at the influence of different wavelengths of light and how the different wavelengths of light influences photosynthetic activity of organisms. So that in itself, photobiology is very complicated but also very interesting. But for me, I'd like to limit my work on the trace metals and the influence of trace metals on these processes. So one cannot do all.
Next question po, another one from an anonymous attendee. Is it possible to do metal analysis from tissues and use it as proxy for the extent of metal contamination in our oceans? Are there bioaccumulated metals that remain toxic except for lead and mercury?
So the first question is if we can use trace metals as a proxy for pollution or contamination. Yeah. So
Is it possible to do metals analysis from tissues and use it as a proxy for the extent of metal contamination in our oceans?
yes, you can. And you can actually monitor metal concentrations up the food chain. So you can look at it from phytoplankton and then the zooplankton and different sizes or species of fish and look at how it is transferred up the food chain. And to do this, you should not rely on total metal concentrations only. You have to look at isotopic fractionation of trace metals because that's very important to look at the isotopic fractionation because that gives you the fingerprint of the metal itself. And not only look at the isotopic fractionation of a specific metal, but also look at concentrations of various metals. So it will be good to, let's say, look at aluminum, look at iron, manganese, copper, blah, 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 and then some of the rare earth elements. So if you have an image of the periodic table in your head, that will be the middle D and the F series in your periodic table. Sorry about that. But yes, so combination of metals as well as the isotopic fractionation of several metals of interest, specifically iron, lead, because lead is important in terms of not just the toxicity, but it's also a very good indicator of anthropogenic input.
Thank you po. Last question po from Zoom so far. From Dr. Jacinto, what analytical methods do you employ to determine trace metals in seawater samples and at concentrations ranging from nano to femtomolar levels? How do you deal with possible contamination associated with water sampling devices?
So ideally, I should have a system wherein I should be able to deploy a Teflon coated sampler, water sampler, and then be able to get aliquots of my water samples in trace metal defined conditions. So a clean container van will be nice. But nowadays, what we are bringing in the field will be our portable trace metal clean bench. So it's really customized. It's just an exhaust fan built utilized in such a way that it's flushing air into the table with passing through a HEPA filter, and then we have a way to have plastic covering all over the place. Okay, so not the best condition, but we do what we have to do, and we make do with what we have. So after that, the sample pre-treatment will be okay. So after acidification, ideally it's passing through different types of resins under trace metal in trace metal clean lab, and then analyzed by ICP-MS. But right now, I don't have all the resins that I need. So what I'm going to do is bring samples to a collaborator in Taiwan and do the sample processing using their CFAST system. So it's an automated system for pre-concentration and purification of the trace metals of interest.
My next question po from Zoom. How has changing conditions brought about by global warming influenced trace metal dynamics in symbiotic dinoflagellates? A lot of the focus is on pH and temperatures, but not so much on the factors that also facilitate or drive physiology.
Okay, so answering this question from the perspective of trace metals. Okay, so right now we're carrying out batch cultures to study the influence of trace metals. But moving forward, what we plan to do is look at trace metals, macronutrient, and all the other growth parameters that we can control, but not just studying the Symbioidiniaceae species on their own, but we'd like to look at this in hospitae. Did I pronounce that properly? Yeah. So within the coral host itself. Okay, so the coral holobiont in itself, that's where we will subject it to varying trace metals, varying nutrients, and all of those conditions in our chemical ecology room, and then look at changes in the coral holobiont as a result of the factors that we manipulated. That's the way that I'm going to approach this. But of course, there are limits in terms of for example, if I want to look at acidification, I don't have that kind of system in place yet.
So, questions from this room? Any more burning questions there? Any last statement from our guest before we say goodbye?
I guess I did say a lot of things already, but yeah. So do check out the lab, and if you have other questions, yeah, you can just shoot me an email. And yes, we can continue discussions.
Well, thank you very much for attending MSI 50 seminar. We really appreciate your interest, your curiosity in what we do and learning about the research that's been done here. Goodbye and have a good day.